Okay, so for the proximal convoluted tubule, 70% of the ions are pulled in there. So um, the red arrow means reabsorption, and this is passive. So you're going to have a higher concentration of these ions or whatnot in the proximal convoluted tubule. They're going to get pulled into the interstitium, so we call that reabsorption. The first time they were absorbed was in the digestive tract. Then they got filtered out at the glomerulus, and now they're getting reabsorbed. Okay, so pay attention to the words, right? So your ions are going to get reabsorbed, water gets reabsorbed, bicarbonate, your glucose and your amino acids. We'll talk about glucose as well because it's a carrier protein. What gets secreted? So secreted is in green, so creatinine, antibiotics, diuretics, and uric acid. So keep in mind, protons get kicked out at the proximal convoluted tubule. The descending limb, what do I want you to focus on? Water. So descending limb, water gets pulled into the interstitium and then into the bloodstream. Whoops. And the ascending limb is going to actively reabsorb or recall sodium chloride. And that actually is going to set up the concentration gradient for water to get reabsorbed. Parathyroid hormone. So for the endocrine lab, we talked about parathyroid hormone. So parathyroid hormone acts on the distal convoluted tubule. When calcium levels are low in the plasma, your parathyroid gland is going to sense that, and it's going to release parathyroid hormone. One of the activities or one of the actions of parathyroid hormone is reabsorbing calcium from your distal convoluted tubule. So calcium gets transported into the interstitium and then into the bloodstream. Distal convoluted tubule is under hormonal regulation, right? So aldosterone is another hormone that acts on the distal convoluted tubule cells. So aldosterone is going to pull in sodium, right? So you're going to reabsorb sodium from the urine into the interstitium and then ultimately into the bloodstream. You don't see the bloodstream here, but it's there. And water is going to follow that, right? Aldosterone, in addition to pulling in sodium and reabsorbing water with it, is potassium gets kicked out, right? Protons get kicked out here and urea gets kicked out here. Then the collecting duct, which is shared by many nephrons, is under hormonal regulation as well, under antidiuretic hormone. So antidiuretic hormone is released when blood particle concentration is high, so greater than 0.9%. So normal physiological range is 0.89 to 0.91, right, percent, or 290 to 310 milliosmoles. That's why we spent time in the beginning of the semester doing the conversion assignment. So anything, let's say 320, 350 milliosmolar, antidiuretic hormone is going to be released from the posterior pituitary, and that is going to target the collecting duct, and you're going to reabsorb water. Okay. I have a separate picture because I wanted to walk through this with you. Let's see. I guess we'll do black and see how that works, right? So at the proximal convoluted tubule, what's reabsorbed, right? So think, actually, let me draw the blood too. Let's draw a blood vessel. Okay, so your blood vessel goes something like this, or your arterial, right? It goes something like this. And then it's going to drop down. If you remember your anatomy, I'm just going to skip that part right there. And then goes back up and out. Okay. So, oops, go away. Okay. So then this goes right there. No, see if I can get this to work. Through here. Ah, sorry, you guys. See if it works good. Okay. Actually, I can move this down. There we go. And then let's go back up here. And this goes down through here, right? And then back up. I can't draw this because all the stuff is in the way. Sorry, you guys. Let's see. Okay, we'll just go right here then. No, maybe we can move this over. Whoops. And that way. There's always something. Okay, and then it's going to go down. And this actually goes back into circulation. So blood flows in this direction, okay? It flows in this direction, then it's going to flow this way. And so this is counter current to the flow of urine in the loop of Henle, okay? You should draw this out, this whole thing, and you should be working on it and understanding what is absorbed where, or what is reabsorbed where, and what effect that has in urine output, right? Okay, so proximal convoluted tubule, let's take it one step at a time. What's pulled in here? So ions are pulled in, right? So remember, it's going to go from the urine filtrate into the interstitium, into the bloodstream. So ions, what else? Okay, good water, gases, H2O, sorry. Looks really bad. Let's see. So water, let's see, CO2, O2, right? Your amino acids and your glucose. So pretty much everything gets pulled in there or gets reabsorbed back into the blood from the proximal convoluted tubule into the blood. Glucose, I want to take a second to talk about glucose. So glucose and amino acids use carrier proteins, right? So this carrier protein is going to pull glucose in. Now glucose is going to go from where it's highly concentrated to where it's lower, right? So glucose gets pulled in to the bloodstream, and it's a carrier protein. So if there's normal glucose in the blood, whatever glucose is in the bloodstream is going to get filtered out, right? And then whatever gets filtered out gets reabsorbed back into the bloodstream. You're not going to pee out glucose because glucose is a high energy compound. You're going to use that for fuel, right? But when somebody has high glucose, as they have diabetes mellitus, high glucose in the bloodstream, they get a lot more glucose filtered out, okay? Way more glucose filtered because there's more glucose in the blood. So whatever is in the blood is going to get filtered out at the glomerulus. And then whatever gets filtered out, normally it gets reabsorbed, right? Gets pulled back into the blood. But there's going to be so much glucose that you can't possibly reabsorb all of that. And so glucose is going to go into the urine and it's actually going to pull water towards it. So if you have diabetes mellitus, glucose is going to pull water back into it, right? And so you're going to have a higher urine output and you're going to have glucose in the urine. Okay. So that's one thing. Another thing that I want to mention is that protons get kicked out at the proximal convoluted tubule. So protons or acid gets kicked out in the proximal convoluted tubule. And so if acid gets kicked out in the proximal convoluted tubule and at the collecting duct, right, what can you expect go down to the bladder? What can you expect for pH? So normal pH should be about 5 to 6. Normal pH range is 5 to 8, but usually it's more acidic. So you're kicking out protons. You're actually reabsorbing bicarbonate here, HCO3. So this goes with the proximal convoluted tubule. And you are synthesizing bicarbonate here. You're not reabsorbing it, you're actually synthesizing it. So the kidney makes or synthesizes bicarbonate. And so bicarbonate is going to, remember it's a buffer, so buffer maintains the pH of whatever solution it's in, so it's going to maintain the pH of the bloodstream 7.35, 7.45, right? Okay. All right, so let's go over proximal convoluted tubule again, because I think I kind of went a little crazy. So you're going to pull in ions or reabsorb ions. You're going to reabsorb water. You're going to reabsorb some gases, right? Amino acids, bicarbonate, and you're going to kick out or secrete protons. One thing that you need to be mindful of is glucose. So glucose is going to get reabsorbed too. But this carrier protein, because it's a carrier protein, it can get saturated. It doesn't in a normal, healthy individual. So if you or I chugged a couple of Gatorades, we would not expect glucose to be in the urine, right? When glucose is in the urine, it's telling me that there's too much glucose in the bloodstream. So too much glucose is getting filtered out. All of it's getting filtered out, right? And then whatever can is going to get pulled back into the bloodstream or reabsorbed, but you can't possibly transport all of that glucose back, so you lose some in the urine. So in a normal healthy individual, do you expect to see glucose in the urine? No, you don't, right? So there should be no glucose in the urine. What if we see glucose in the urine? That means a person has diabetes mellitus, but I need you to be more specific. It means that they have diabetes mellitus, so that they have high blood sugar, and in the proximal convoluted tubule, those carrier proteins are becoming saturated. And when they're saturated, they're still pulling glucose in, but they can't pull all of it in, and so some gets lost. And so diabetes mellitus, hallmarks of diabetes mellitus, let's put it over here on the bottom left-hand side, diabetes mellitus, right? Glucose in urine. You should be able to explain that for lab, right? What happens to urine output? Because you have glucose in the urine, it's going to pull water towards it. So your urine output's going to increase. And because you're pulling water from your blood into the urine, what happens? Good, you're dehydrated. So if you're dehydrated, what does that give you? Thirst, right? Okay, good. Okay, so we'll come back to that if we need to. All right, so basically how the glomerulus works is whatever's in the blood gets filtered out, except for, remember, your large plasma proteins. So, or your large molecules like your red blood cells, white blood cells, your platelets, and your albumin, right? Your large plasma protein. So all of those stay stuck in the bloodstream, and everything else gets filtered out. So your glucose, your water, your ions, your bicarb, your urea, your creatinine, your amino acids, what have you, everything else, hormones, right? So pregnancy hormone, human chorionic gonadotropin gets released, and so you can test that in the urine. Ketones, right? So metabolic excess of metabolites like ketones, waste products. Okay, so your proximal convoluted tubule reabsorbs most of the stuff, right? Let's take a look at the distal convoluted tubule, and then I'll come back and talk about the loop of Henle. So I want to do distal and collecting duct. So distal convoluted tubule is under hormonal regulation. So when you have low blood calcium, what hormone do you release? Good. Parathyroid hormone. Whoops. And that's going to cause you to pull in what? Or reabsorb calcium. Good. Okay. So parathyroid hormone causes you to pull in calcium. What's another hormone? So when you have low blood volume is going to cause you to release aldosterone, right? So aldosterone does what? Aldosterone is going to cause you to pull in sodium, and I would write it in a different color, and what's going to follow it? Good. Water is going to follow it. Also, I want to add to that you kick out potassium, right? So you kick out potassium because of aldosterone as well. So when do you release aldosterone? You release aldosterone when blood volume is low, which causes a drop in blood pressure, right? Or MAP. Or when you have low sodium in the bloodstream, right, sensed by the macula densa cells, or high potassium. Remember, we really want to regulate potassium in the plasma because high potassium or low potassium is going to affect excitable cells' resting membrane potential. And so we really want to regulate potassium, and so aldosterone is going to do that. Some diuretics can actually throw sodium levels off and potassium levels off. So you want to be able to measure or keep an eye on potassium. Okay, so let's go to the collecting duct. So collecting duct is where you kick out protons from the blood into the urine filtrate, and you reabsorb bicarbonate. So bicarbonate ion is pulled into the bloodstream. Or not reabsorb, I'm sorry, this is actually where you synthesize. Whoops, sorry you guys. It's where you synthesize bicarbonate. Okay. All right, another thing that goes on in the collecting duct, let me see if I can erase it. Another thing that goes on in the collecting duct is that you oops. Get my pen back. Is it's under hormonal regulation as well. So when you have high particle concentration of blood or high plasma osmolality, what senses that? Your hypothalamus does, right? And that's going to cause your anterior pituitary to release what hormone? Good. So you increase antidiuretic hormone when you have high plasma osmolarity. Now this could be because you're dehydrated, right? You've sweat, you've lost water, you have more plasma proteins because you've lost the water. This could be because you ate a bag of potato chips. This could be because you have glucose, excess glucose in the bloodstream, right? So all of those contribute to plasma osmolarity. Be careful with dehydration because dehydration is going to trigger this first, and then it's going to trigger aldosterone release because blood volume is going to drop. Okay, so in the early stages of dehydration, your particle concentration is going to go up, or your osmolarity of your plasma is going to go up. And so it's going to trigger antidiuretic hormone release. In the later stages, blood volume is going to drop. So blood volume is going to drop, blood pressure is going to drop. So if somebody is dehydrated, blood pressure is going to be low, and then heart rate's going to be high to compensate for that. And they're going to be releasing aldosterone, and they already have been releasing ADH. Okay, so let's go back to the ADH pathway. So increase in plasma osmolarity, increase in antidiuretic hormone. ADH is going to go to the collecting duct and actually cause you to insert these molecules called aquaporins. So increase aquaporins. And you're going to pull water back into the bloodstream. Okay, so you're going to reabsorb water. So if you're dehydrated, you release ADH, or if you've eaten, a bag of potato chips or French fries, or you have diabetes mellitus. There's various reasons, but that's going to increase plasma osmolarity. You're going to release ADH, and that's going to cause you to pull water in. Excuse me. From the urine into the bloodstream, so that you actually decrease plasma osmolarity. Okay, the line of thinking here is if you're pulling water back in or you're recalling water, what happens to water getting kicked out? It drops, okay? So if you are releasing ADH, if you have a high plasma osmolarity and you're releasing ADH, you're reabsorbing water. And if you're reabsorbing it, you're not peeing it out, right? So there's two things you can do with water that you filtered out. You can reabsorb it back into the bloodstream, or you can pee it out, right? So if you're dehydrated, you guys know already that you're not going to urinate it out. You're going to reabsorb it. And so what happens to urine output when you're dehydrated? It drops, right? So let's look at ADH. What does ADH stand for? Antidiuretic hormone. What does a diuretic do? A diuretic makes you diurese, right? Pee more. Antidiuretic hormone is going to make you pee less or diurese less. And so why? Because you're reabsorbing it back into the bloodstream. Okay, so for urine output, normally urine output is about 100 mils, right? It really depends on your level of hydration. So we'll just say about, 10 to 100 mils, right? Specific gravity is the particle concentration of your urine. So specific gravity is how many particles. So if you go back up to the glomerulus, you're always going to be dumping out particles, right? So there's always a set amount of particles that is going to be in the urine. What changes is the amount of water that's in the urine. So if you're dehydrated or blood volume's low, you're going to not pee out the water. You're going to reabsorb it. And so your urine concentration is going to be high. And if your urine concentration is going to be high, your specific gravity is high. So if you're really hydrated, your particle concentration in the urine is going to be low because you have more water. And so specific gravity is going to be low. And so specific gravity is usually around, let's say normal is 1. It's a really weird number, 1.010. So we call this 1010, right? Or 1.010 is actually the proper way to say it. But, in nursing or in the ICU, you're going to say 1010 or 1030 to denote the specific gravity. So the units is units of gravity. Okay. We also use specific gravity for saline solutions, or like when I did shark research and I was interested in uh, how uh, how salty the water was or what the salinity of the water was, and so we looked at specific gravity as well. Okay, the idea behind this is that specific gravity of water is 1.000, and the more particles you have, the higher the specific gravity is. So specific gravity can range from 1.001, which is really close to water, to 1.030, which is really concentrated. All right, so take a second to think through these. Oh, let's talk about the loop of Henle. Okay, so the loop of Henle is where we have, it's really interesting. It's called a countercurrent mechanism. So what ends up happening, the short version is that we are going to reabsorb sodium. We reabsorb a lot of sodium down here. So I'm going to make a big arrow. So reabsorb, a lot of sodium here, and it's active. Then we pull in a little bit less here, and then we pull in a little bit less here. And when I say pull it in, I mean in all areas, right? So we're actively pumping it from the ascending limb into the interstitium and ultimately into the bloodstream. Okay. So the blood, let's take a look at your bloodstream. So this is 300 milliosmoles, right? And as we go down and we're pulling sodium in, what happens to the concentration? So concentration goes from 300, let's say 600 milliosmoles, 900 milliosmoles, and then 1200 milliosmoles, right? And then this is 1200 milliosmoles, and this side is permeable to water. This side is not permeable to water. So the only way that this works is if this side is not permeable to water. Okay, water can't get through. So you're pumping sodium out to make a lot of sodium, to have a lot of sodium into the blood, or in the blood, I should say. And that is going to cause water to move into the bloodstream. And so, because this is more particles, so water is going to move into the bloodstream. Water is going to move into the bloodstream. And water is going to move into the bloodstream. This is a really effective way of getting water to move into the bloodstream. So if this is 1200 milliosmoles and water moves into the bloodstream, then it becomes around 900 milliosmoles, water moves 600, and then water moves 300. And so by the time you're done with this, you started at 300 and then you end up at 300. Animals who live in the desert, like kangaroo rats or camels, have a really, really long loop of Henle so that they can maximize water reabsorption. A lot of the water is pulled into the proximal convoluted tubule, but don't underestimate this loop of Henle. Okay, so another thing that I wanted to tell you is, you notice these lines here. This is the cortex, right? So here is our cortex. And I'll put it over here. Oops, go away. Okay, so here's our cortex. What is the concentration of the cortex? Of the kidney? It's around 300 milliosmoles, okay? So everything in this whole region is 300 milliosmoles. What about the medulla? So this is your medulla, right? You know that the loop of Henle extends down into the renal medulla. The medulla gets more and more concentrated. So medulla goes from 300 to 600, right? And there's actually gradients. So it goes 600, whoops, sorry, you guys, I got excited. 600 and then 900, you get the picture, and then 1200, right? So as you go more deep into the medulla, you see an osmolarity shift. You see the tissue get more and more concentrated. How does it get more and more concentrated? You actively pump sodium into the interstitium and into the bloodstream. Likewise, the collecting duct is actually kicking out urea as well. So urea is kicked out here, and then it's pulled back in here. And so you have this amazing concentration gradient. So let me recap. So as urine flows through the descending limb of the loop of Henle, water gets pulled into the interstitium and into the bloodstream. The further or the more deep you go, or the further down you go into the loop of Henle, the more water gets pulled out. And so what would you expect to see for urine concentration right here? Good, your pre-urine concentration is around 1200 milliosmoles. Really concentrated, okay? And then you come back up, and at the ascending limb, you're kicking out sodium. And so as you're kicking out sodium, what happens to the concentration of the urine? Good, it gets less and less concentrated, or more and more dilute. And by the time you come up to the cortex, it's actually 100 milliosmoles. So you started at 300, and then it got super concentrated, and then it got really dilute. And it's actually 100. And then once it hits the collecting duct, depending on how hydrated or dehydrated you are, it can get really concentrated again. So it can stay at 100, it's usually around 300, but it can go up to 1200, okay? So this would be really concentrated urine. That would be your dark yellow urine if you're really dehydrated, because you're letting go of the particles, but you're pulling water back into the bloodstream, or it can be really dilute. And so if you're really hydrated, if your body's hydrated, you're going to dump water into the urine because you don't need it. And so it's going to be really light colored. Okay. All right. So I want you guys to practice drawing this out and understanding what gets reabsorbed where and how that gives us our urine parameters. Okay, so I want to show you, I'm not going to ask this on the exam, but I want to show you what this looks like. So this is, let me see where my picture, there we go. So this is sodium reabsorption in the proximal convoluted tubule. This is active transport, right? So notice here's an interstitium. Let me orientate you. Here's an interstitium. Here's the basolateral membrane of the simple cuboidal epithelial cell. Here is the apical membrane of the cuboidal cell. So notice that you have a sodium-potassium pump. It's going to kick out 3 sodium. So sodium's high in the interstitium and drag in potassium, 2 potassium. So this makes sodium low in the cell and sodium high on the outside. So now that sodium's low, which way is sodium going to go? So the urine filtrate sodium is going to go from where it's high to where it's low. So the sodium potassium pump, and I told you we would see this again when we get to urinary system or kidneys, the sodium potassium pump is kicking out sodium so that sodium's low here, and then that's going to aid sodium going into the from the urine into the cuboidal cell. Okay. Whoops. Glucose. So glucose, and so you can add to your picture for glucose. So let's talk about glucose reabsorption in the proximal convoluted tubule. We already talked about it, but glucose is freely filtered at the glomerulus. So this means whatever glucose is in the blood gets filtered through the glomerulus, right? And whatever gets filtered at the glomerulus, it gets reabsorbed in the proximal convoluted tubule. So normal glucose levels, right? So around 70 to 100 um, milligrams per deciliter, right? Um, gets filtered out. So whatever's in the bloodstream is going to get filtered at the glomerulus, and whatever's at the glomerulus is going to get reabsorbed. So normally, in a healthy individual, no glucose should be in the urine. So if I drank a gallon of Gatorade or a couple gallons of Gatorade, all that glucose is going to go into my bloodstream, and it's going to get filtered out of the glomerulus, and it should get reabsorbed in the proximal convoluted tubule. Therefore, I should not have any glucose in the urine. If I do have glucose in the urine, what is that a sign of? Good, that's a sign of diabetes mellitus. That means that I had high glucose in the bloodstream and all of it was filtered out, and the carrier proteins got saturated, and so I couldn't reabsorb all of that in the proximal convoluted tubule. And so I left some glucose in the urine, right? Here's a picture. So here's your sodium potassium pump. So you're kicking sodium out, sodium's low here. Sodium is going to go from the urine into the cell and then into the interstitium, right? So urine's going down its concentration gradient and it's dragging glucose with, right? So it's dragging glucose with, and then glucose is going to go from where it's highest to where it's lowest. So it's going to go into the bloodstream. Pretty nifty, huh? This guy gets saturated, right? And so when there's too much glucose, this carrier protein can't pull all that glucose in as fast as you need it to. And so glucose is left in the urine. Normally, this guy plugs and chugs and it's fine. But when you have high blood sugar, in the case of diabetes mellitus, you have too much glucose, and all that glucose can't possibly be reabsorbed. This shows plasma concentration of glucose and rate of glucose movement in milligrams per minute. So notice that filtration, the filtration line slope doesn't change, right? So this means that at 100 units of glucose, you're going to get about 100 milligrams per minute. 200 units of glucose, 300 units of glucose, 400. So whatever, this is telling me whatever the plasma concentration is of glucose, I'm going to get that filtered out. For normal concentrations of blood glucose, whatever is filtered out is reabsorbed. You notice how these lines both match, the blue one and the purple one, right? But once I have too much glucose in the bloodstream, that's going to get filtered out, but then those carrier proteins are going to start to get saturated. And so what you're seeing a slow decrease in reabsorption. And if you're not reabsorbing it, what are you doing with that glucose? You're kicking it out, excreting it, right? So around 180 is your renal threshold, right? So that's when you stop reabsorbing or start to decrease reabsorption and start to kick out glucose in the urine. So notice that if somebody has blood glucose level of 300, which is not normal, they have plateaued in reabsorption and they're now peeing out whatever glucose is extra. Okay. My father has diabetes mellitus, and his blood sugar before he was diagnosed was around 600. And so he was excessively thirsty, excessively urinating out, because he had so much glucose that he was kicking out in that urine, or so much glucose that was getting released in the urine at 600 milligrams per deciliter, right? You're excreting out a lot of glucose and you're reabsorbing. Your transport maximum is at 300, about 380 milligrams per minute. So you can imagine how much glucose was in that urine. It's like sugar pee. Okay. We can skip this. So cells in late distal tubule and collecting duct regulate balance. So principal cells, you have two different types of cells in that epithelial lining. The principal cells pull in water and electrolytes, and then the intercalated cells deal with acid-base regulation. So here's the ion reabsorption. So sodium reabsorption is coupled to kicking out potassium. It's also coupled to kicking out H+. Aldosterone increases sodium reabsorption in the distal convoluted tubule. Aldosterone is a steroid hormone. So if it's a steroid hormone, it is going to go through the plasma membrane or not. Okay, we'll find out. It's secreted from what zone? So it's secreted from the adrenal cortex, the zona glomerulosa. You should know that, right? So you need to start working on drawing out your homeostatic feedback loop for RAS and the aldosterone part. You can use your endocrine lab to do that. The aldosterone acts on principal cells of the distal tubules and increases the number of sodium potassium pumps and open sodium and potassium channels on apical membrane. Let's take a look. Potassium balance, we'll look at that in a second. Sorry, potassium balance. So aldosterone is a critical hormone in regulating plasma potassium in narrow range. Plasma potassium is around 3.5 millimolar to 4.5 millimolar. And so you want to, aldosterone, if potassium is high, aldosterone is going to increase and kick potassium out into the urine. You don't want hypokalemia, which is low plasma potassium, because your excitable cells are not going to hit threshold. They're not going to depolarize. Hyperkalemia is excessive potassium, and that means that your excitable cells are going to depolarize faster because they're closer to threshold. Okay, causes include kidney disease, diarrhea, and diuretics. You want to pay attention when somebody's on diuretics, what their potassium level is for the blood. Okay, let's take a picture of what happened, or excuse me, let's look at the picture of what happened. So when blood volume drops, which causes a drop in blood pressure, your RAS system gets activated. The end result is releasing aldosterone into the bloodstream from your zona glomerulosa of your adrenal cortex, right? Aldosterone is going to go into the cell, bind to a receptor, cause you to increase sodium potassium pumps on this side of the membrane, and cause you to increase sodium and potassium ion channels on the apical membrane. So sodium is going to get pulled in faster and potassium is going to get kicked out faster as well. So really interesting. And keep in mind, this is a steroid hormone. So if it's a steroid hormone, it can go right through that plasma membrane. Okay, acid-base balance. Normal pH of arterial blood, 7.35, 7.45. You know that if pH is low, right? So if it's more acidic, you get conformational changes in your protein structures, most notably in hemoglobin, and it affects how hemoglobin binds oxygen. You change your excitability of your neurons, changes in potassium balance, really dangerous, right? Can lead to cardiac arrhythmias, vasodilation of blood vessels, etc. So we really want to regulate your blood pH. So inputs of acid, your diet, your proteins and fats through metabolism. Well, proteins and fats, breaking down proteins, are going to give you protons, right? For metabolism, CO2, lactic acid, and your ketones, your keto acids, are going to increase blood H plus as well, or blood protons, right? So acid, if you will. How do we deal with that acid? We can regulate blood pH or acidic blood by kicking out CO2 with the lungs, right? So if you're making more CO2, you can kick it out of the lungs, or you can kick it out by H plus, or kick out the protons of the kidneys. So we're not going to get into the metabolic or respiratory acidosis or alkalosis here, but if you can understand that from breaking down proteins and fats, we get protons, and from CO2, lactic acids, and keto acids, we get protons as well. How do we, and that's going to drop the blood pH. How do we handle that? We're going to kick out the protons at the kidneys, and we're going to kick out CO2 at the lungs. And so there is a metabolic way to handle it, like excreting it, or there is a respiratory way to handle it as well. How do we handle hydrogen and bicarbonate? So the proximal convoluted tubule is where bicarbonate is reabsorbed and you kick out protons, okay? So hydrogen ion, you can put proton there. So bicarbonate's reabsorbed. Bicarbonate is gonna act as a buffer, so you're gonna reabsorb that back into the bloodstream, and protons are gonna get kicked out. Your distal tubule and collecting duct is where you actually secrete hydrogen ions, and you're going to synthesize new bicarbonate ions. So your collecting duct is where you make bicarbonate ions. You actually synthesize them and pull them into the bloodstream. And so the kidney does regulate pH as well. You don't have to know it, but just so you can appreciate it, right? Your CO2 gets pulled in, combines with water, hits carbonic anhydrase, makes carbonic acid. That is going to dissociate into protons and bicarbonate. You're going to pull bicarbonate into the peritubular fluid, or the interstitium. The H plus is going to get kicked out, so H plus gets kicked out. And that H plus is going to keep binding to whatever bicarbonate is in urine and pull it in. So H plus gets kicked out, and that H plus is ultimately helping pull bicarbonate in so that you can keep the bicarbonate in the bloodstream. Notice how it's carbonic anhydrase. So you see carbonic anhydrase here, just like you did when we talked about red blood cells. Hydrogen ion secretions, here's another way hydrogen ions are secreted. So they combine, the hydrogen ions are kicked out, they combine with HPO4 to make H2PO4. Let's talk about urine concentration. So how does a urine change as it flows through the nephron? So again, this top portion is the cortex. So this is a more superficial part of the kidney. And then this bottom portion is the medulla. So we're going more deep into the medulla. So the cortex is around 300 milliosmoles, okay? So the urine filtrate, right, because it came from the blood, is around 300 milliosmoles. So blood is 300 milliosmoles, urine filtrate is around 300 milliosmoles, okay? Notice how when we hit the, when the urine, pre-urine, or the urine filtrate, same thing, goes into the loop of Henle, the descending limb of the loop of Henle, you're going to pull water from the loop of Henle into the interstitium. Why do we pull in water in the descending limb of the loop of Henle? Because we had this concentration gradient set up by the ascending limb of the loop of Henle. So number 2, ions are reabsorbed, but water can't go through. It's impermeable to water. So we pulled in a bunch of sodium from here all the way up, right? So that sodium would stay in this interstitium in blood and pull water in. So really, it's really actually really cool, you guys. So the ascending limb actively reabsorbed sodium from the urine into the interstitium to the blood so that we would be able to pull water in on this side. So and it's really sort of counterintuitive. You expect sodium to pull in here and water to pull in here, but it doesn't happen that way. This sets up the concentration gradient to pull water in on this side. Take a look at the concentration gradient. So this is 300 milliosmoles. Then this is 600 milliosmoles, 900, and 1200 milliosmoles, right? At the distal convoluted tubule, your urine is about 100 milliosmoles. And then when it hits the collecting duct, as it goes more deep into the medulla, you're going to pull water in or reabsorb water depending on how much ADH is present, right? So permeability to water and solutes is regulated by hormones. So ADH, if you're really dehydrated, you're going to have a lot of ADH and you're going to reabsorb a lot of water so that your urine is really concentrated, right? Why is it concentrated? Because you're not peeing out the water, you're reabsorbing it. If you are really hydrated, right, let's say I've been drinking enough water, you're not going to have a lot of ADH here. And because you don't have a lot of ADH here, you're not going to reabsorb water. And so you're going to pee it out. And so your urine particle concentration, it's going to be around 50 milliosmoles. Good. Here's another picture. So just reiterates the idea. Water is going to get pulled into the interstitium and into the bloodstream. Look at the concentration gradient, 300, 400, 600, 900, 1200. And then urea and sodium are going to set up that concentration, right? So urea is getting pulled out of the collecting duct and sodium chloride is getting kicked out of the loop of Henle. And so the sodium and the urea actually make the concentration gradient. So they make the 1200, they make the 900 here, the 600 here, the 300 here. And the red arrow means active transport. So you're actively kicking out sodium there. And then you're pulling water out in water and urea. So this is really interesting. Countercurrent exchange. So let's add the blood vessel to it, right? So here's your loop of Henle. So you're pulling water into the interstitium, and then you're actively transporting sodium into the interstitium. So what ends up happening? Sodium gets pulled into the interstitium or kicked into the interstitium. It's going to go into the blood. It's going to go into the vasa recta. And notice the vasa recta flows in the opposite direction as the urine does. Whoops. And so you're kicking sodium in, sodium, sodium, sodium. You're making it more and more and more concentrated so that when the blood gets to this side, when it turns around, you're going to pull in water. You're going to have a really strong concentration gradient to pull in water, okay? This is called countercurrent exchange or countercurrent mechanism. And this helps pull in a lot of water. Notice that the blood osmolarity hasn't changed. It was 300 at the top of the loop of Henle. It got super concentrated, and then you pulled in a lot of water. So add salt, 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 sodium, 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 water, water, water. And then it's leaving at 300 milliosmoles. The urine is 300 milliosmoles, goes to 600, 900, 1200, and then back up to 100. Notice that this is telling you that water can't move, right? So this side is impermeable to water movement. There, you can look at a countercurrent exchange animation here. Take a look at that one. The last thing that we need to talk about is water reabsorption in the collecting duct. So notice that the collecting duct goes through the renal medulla, right? Renal medulla gets more and more concentrated as we go more deep. If there's no ADH present, which never happens, but if there's no ADH present or less ADH present, you're not going to have much water getting pulled in or reabsorbed, and the urine's going to be really dilute, okay? So this urine being really dilute, what is the specific gravity of that? Good. So dilute urine has a specific gravity, so Sg. So it has less particles, so it's going to be 1.0, let's say 0.5 units of gravity, okay? So if somebody's really hydrated, they're going to pee out more urine, so hydrated, right? So they're going to increase urine output. And the color of the urine, what is that going to be? They're kicking out more water, so it's going to be a lighter color, right? So clear to light yellow. And we know that the specific gravity is low because there's less particles and more water. So the urine's going to be dilute. Okay, so you increase urine output, specific gravity drops. So the more hydrated you are, the greater your urine output will be in a normal healthy individual, and the lower the specific gravity can be. So you can say that when you're hydrated, urine output increases and specific gravity drops. They're inversely related. Okay, did you catch that? If you're hydrated, your urine output increases because you're not reabsorbing it, you're peeing it out, and specific gravity drops because you're diluting all of those particles in the urine. So they're inversely related. Okay, what happens when you are dehydrated? When you're dehydrated, you release a lot of ADH, right? So increase in ADH when you're dehydrated. And that is going to cause you to pull water from the urine into the bloodstream. And so if you're dehydrated, let's write it over here, right? So you're going to have a lot of ADH. And when I say dehydrated, anything probably around 350 milliosmolar, right? So increase ADH, you're going to increase water reabsorption. And so if you're pulling it back in, are you going to pee it out? No. So what happens to urine output? Urine output drops. Good. And what happens to the particle concentration of the urine? Good. The particle concentration or specific gravity goes up, right? Because you have less water, so specific gravity goes higher. Good. And so it's going to look really yellow, right? So the more dehydrated you are, the more yellow the urine is going to be. Awesome. Okay, let's look at ADH pathway. And we covered this in endocrine and in blood pressure regulation, right? But let's go over it again. So when you have an increase in plasma osmolality, right? So high sodium concentrations and plasma osmolarity, again, 320 to, I think that other feedback loop said 400. Sorry, that's really bad. That's going to stimulate the osmoreceptors in your hypothalamus. So these osmoreceptors in the sun region get excited. Okay, and remember, they are nervously connected via nerve axon to the posterior pituitary. So your stimulus is your osmoreceptors in your hypothalamus. Your integrator would be hypothalamus / posterior pituitary. Posterior pituitary is going to release ADH into the bloodstream. ADH is going to go to the collecting duct of the kidney, so that's your effector, and it's going to cause water to be reabsorbed. That's your response. So water is being reabsorbed. That means urine output drops, right? So you have a drop in urine output. And because you have more particles, what happens to specific gravity of urine? More particles. What color is it? Dark yellow, right? And so specific gravity is going to go up. The amount of particles, the higher the particles, the higher the specific gravity, right? And so it'll probably be closer to 1.030, and it might even be 1.030. Okay. So if you're reabsorbing water, you're not peeing it out. If you're not peeing it out, urine output drops. The amount of particles, because there's less water, there's technically, well, theoretically, I guess, more particles. That's going to be dark yellow, and so specific gravity is going to increase. If you're reabsorbing water, what happens to plasma? Plasma volume goes up and plasma particle concentration drops. So you're diluting plasma, right? So because you're reabsorbing water, you're diluting the particles in the blood, and so that is going to decrease the concentration of the blood, right? So you need to think about the kidney. So the kidney's job is to, or the nephron's job is to filter out everything, well, almost everything, right? And then reabsorb whatever you need or recall whatever you need. If you are dehydrated, you're going to pull in or recall more water and not pee it out. If you're hydrated, you're going to pee it out and not recall it. Okay, and so you need to think of it from that standpoint. You can look at someone's urine and you can assume if it's dark yellow and there's not much urine that they're dehydrated, right? Or you can assume if it's dark yellow, they're going to have a high specific gravity. You can also assume by looking at urine that if it's dark yellow, they probably have a lot of ADH in the bloodstream, right? Okay, good. How does ADH work? You have ADH in the bloodstream. It's going to bind to receptors on the surface of the cell. So is it a steroid hormone or a protein hormone? Good, it's a protein hormone or a peptide hormone because it can't get into the cell. It has to bind to a receptor on the surface of the cell. I'm at number one. And then it's going to turn on a G protein mechanism, which is going to activate cyclic AMP, protein kinase A, and cause you to merge this vesicle full of aquaporins or exocytose this vesicle of aquaporins into the plasma membrane. So you get more aquaporins on that apical membrane. More aquaporins, more water getting pulled into the cell and then into the bloodstream. Pinky. Okay, so how do we regulate blood pressure? We've talked about this for several weeks now. So let's go back to homeostasis. Oops. Normal blood pressure, right? So if blood pressure drops, whether or if there's high body temperature, trauma, if we have increase in CO2 or prostaglandins or increased activity of the tissue, whatever it might be, that is going to cause your blood vessels to dilate and decrease resistance, and homeostasis should be restored at that local level. Let's say I'm doing bicep curls, okay? I'm gonna get more blood flow to the biceps, and that should regulate it. But what if that doesn't work, right? So if autoregulation is ineffective, so let's say I start exercising, right? Local level is not going to provide enough regulation. So then it's going to be nervous system. So you're going to do your baroreceptor reflex, right? So baroreceptor reflex is going to regulate blood pressure. And what if the baroreceptor reflex doesn't regulate blood pressure? Like let's say I am dehydrated and blood volume is really low, right? Because I've lost a lot of water to the environment. Let's say I'm hiking out a Joshua tree in the summertime. And so I go through the baroreceptor reflex and my blood pressure is still low. Then what? Then it's going to be hormone regulated. So you're going to release aldosterone, and that should restore blood volume. If aldosterone doesn't restore blood volume, then you've got a problem. Then you're going to go into heat, shock, and it's going to be a disturbance. You need to get to the ER and get fluids quickly. Okay, so for your analysis, so for the urine lab, if we met in the urine, well, let me rephrase that. If we met on site, you would actually pee in a cup and analyze your urine, right? So when we analyze our urine, we are looking at urine output, right? So urine output. What are normal values, right? Of course, you want to know, whoops, sorry guys, I didn't mean to cross it out. You want to know normal values, and you want to know how they change when you drink these different solutions. Actually, let me see if I can, well, whatever. No, I want to see if I can erase it. Sorry, you guys, I struggle with being a perfectionist, surprise, and wasting time erasing everything and redrawing it. Okay, so let's see. Okay, so normal. You want to know what normal levels are for urine output. You want to know what normal level is for specific gravity. You want to know what urine pH is, right? You want to know if we can expect to see glucose in the urine, and you want to know if we can expect to see proteins in the urine. Ketones, right? So if we skip breakfast and lunch, or we're fasting, or we're not eating enough, you're going to see ketones in the urine because you're breaking down fats, you're going through beta oxidation. Do we see albumins in the bloodstream, right? Or proteins? I guess I should say proteins. So we normally don't see proteins in the urine, but if you have a kidney infection where you have bacteria eating away at the glomerulus, you're going to kick out proteins when you're not supposed to be doing that. Or dehydration or stress, you're going to kick out albumins. Should we expect to see bacteria? No, right? Should we expect to see red blood cells? No, but if you have a urinary tract infection, you're going to see red blood cells. Likewise, you're going to see bacteria, you're going to see white blood cells, and you're going to have nitrites because the bacteria make nitrites as a waste product. Okay. There's urobilidrin, there's crystals. We could keep going with this list, right? Actually, my baby was recently dehydrated. He got his 6-month shots, and he was not a happy camper, so he wasn't drinking enough, and his diaper was orange. So he was peeing out crystals because he didn't have enough water, and so he was making crystals. It went away after 24 hours, so it was actually one time that he had crystals in his diaper. But so you can tell a lot about what's going on by analyzing somebody's urine and by analyzing somebody's feces. Okay, so let's go back to this. I want you to know normal ranges for urine output for urine, right? Specific gravity, pH, glucose, protein, ketones. All of these you should not see, right? You should not see them in a urinalysis, we call it, or a UA, right? Should we expect to see glucose in a normal, healthy individual urine? No. What should pH be? Around 5 to 7, right? So normal about 6. Specific gravity is going to vary. It's about 1.001 to 1.0, let's say 10. If you want to go with an easy number, 1010 is normal. Urine output, it depends, about 50 mils to 100 mils, right? Per, I don't know, hour, 2 hours, etc. So you want to know what normal parameters are for yourself, for other people. If we did this in lab, you would test yours using a urine dipstick and match up the colors on the urine dipstick with the colors on the container, excuse me. And you would know what your urine output was, specific gravity, pH, okay? Then I would have some people drink excess water, so 500 mils of water, or 500 mils of sugar water, or 500 mils of bicarbonate, which is is a buffer, but bicarbonate, excess bicarbonate is going to increase the pH of the bloodstream to 8. Okay, so that's going to affect your pH of your bloodstream. So you're going to hold on to protons, right? Salt water is going to dehydrate you. aldosterone and ADH. So you shouldn't be prepared to discuss these for lecture and for lab. Figure out what normal is, right? And I gave you the data. So I gave you data on the urinalysis lab so that you can figure out what normal values are. So normal values would be your no treatment person, right? Your no treatment person are going to give you your normal values. And then I had data from somebody who drank excess water, somebody who drank sugar water, someone who drank sodium bicarbonate together, so it's going to dehydrate you and shift the pH, the real kicker. And then what if I injected aldosterone into somebody, or what if I injected ADH into somebody? How would all these parameters change? All right, that's your goal. Okay, so I want you to work through these. And then take some time and work through these. So stop the video, take some time and work through these, and then we'll go over these, okay? The last thing is micturition. So micturition is the storage of urine and the micturition reflex, okay? So here's the bladder. So notice you have a motor neuron that's firing, that's constricting this external sphincter. This is skeletal muscle sphincter. And then the smooth muscle sphincter, the internal one, is passively contracted. So as the bladder is filling, how do we not pee out stuff, or how do we not urinate? That smooth muscle is contracted, right? So the more this stretches, the more the smooth muscle contracts, and then this is contracting using a motor nerve. When the bladder gets super full, when it gets super full, the stretch receptors in the bladder lining send a signal to the parasympathetic nerve to constrict smooth muscle of the bladder and shut off this motor nerve so that this external sphincter relaxes. And so once your bladder gets too full, it causes contraction of the smooth muscle and it causes relaxation of that sphincter. You can imagine in, kittens, puppies, babies, how hard this is. So if you have a toddler in your potty training, you're trying to get them to understand their brain, to understand that sensory nerve impulse, right? So when their bladder gets too full, that's sending a signal to your brain, hey, I have to go to the bathroom, hey, I have to pee, hey, I have to urinate. And then eventually, in keeping this motor neuron constricted, this external sphincter constricted until this shuts off and relaxing this. When you put a Foley catheter in somebody, the catheter sits above this sphincter and it drains out the urine. And so this bladder never stretches. It never gets full enough to send that stretch receptor, get that stretch receptor excited. So when you take out a Foley catheter, you have to retrain the bladder. So the stretch receptor, what they usually do is when they know they're going to pull out the Foley catheter, they'll actually clamp the line so that the bladder fills. So the stretch receptor triggers this action potential that causes the motor neuron to stop firing and causes a smooth muscle to contract. The catheter will still be there. So when you pull the catheter out, this sphincter needs to be retrained to stay shut. And it takes a little bit of time, a couple of weeks. But it eventually will get there. Okay, let's see. Okay, so I'm going to exit this. Let's see. Discard this because we have it already. I am going to go back to, let's do a whiteboard drawing, you guys. So, and this will just be 10 more minutes. So let's do whiteboard. Okay, so let me move this over. Okay, so I want you to draw out your nephron, and I want you to draw out what is reabsorbed at each step, right? So the first thing that you need to do, and if we were in lab, I would cover this again, but you can go back and listen. Nephron. I want you to know what's reabsorbed where for lecture, what's kicked out, right? How do we regulate, glucose? How do we, the whole aldosterone pathway, ADH, urine output, et cetera, dehydration. But for lab, what we would have done is we would have predicted what would happen when you drink different solutions. So we would have covered the solutions, right? And then the urine output, the specific gravity. Whoops, sorry guys. pH. I guess I could have made this bigger. Let me make this bigger, guys. Sorry. Okay. And I want you to look at the review sheet. So you need to spend a lot of time understanding this because this is not something that's going to come easy. Oops, I want to draw. There we go. Okay, so let's try this again. So solution. Then let's see, urine output. So urine output, specific gravity, pH. What else am I missing, you guys? Urine output, specific gravity, pH, glucose. So should we see glucose? Protein. Okay, so let's go through and figure out our predictions for this. So solution one would be no treatment, right? So we would have somebody who did no treatment. What would urine output be? So normal urine output, this is where you need to look up normal ranges. So normal urine output would be, let's say 100 mils, right? Usually about 10 to 100 mils an hour. Specific gravity, normal specific gravity is 1.010 units of gravity. pH, let's say pH 6. There should be no glucose, no protein, right? What if someone drank 500 mils of water? 500 mils of water. So they drank 500 mils of water. Let's say they're already hydrated. What would happen to urine output? Urine output would increase, okay, because they're already hydrated, so they are dumping water in the urine. What would happen to specific gravity? Specific gravity would drop, so that means it would get closer to 1.000, which is 1.001. Is water going to affect the pH? Nope. Should you see glucose in the urine? Nope. Protein? Nope. Okay, then somebody is going to drink, another person would have drank 500 mils of sugar water. So let's do glucose plus water, okay? So you're still drinking water, so urine output's going to increase. Specific gravity is going to drop. pH is going to stay the same. If we drink glucose, should we expect glucose in the urine? No, okay? Just because we drink glucose, our body should process it. So we would release insulin and pull glucose into the cells. No protein, okay? What about 500 mils of, in lab, it would have been sodium bicarbonate. So it would have been 500 mils of sodium bicarbonate or baking soda water, which is really gross. It kind of tastes like Alka-Seltzer. Let's separate them though. So let's look at sodium water, right? So if we have more salt, what's going to happen to blood? Blood particle concentration or osmolarity is going to increase. And so we're going to hold on to water. So urine output's going to drop. And because we're peeing out less water, same amount of particles, specific gravity is going to go up. Are we screwing with pH? No, we're not. Whoops, so it's going to stay 6. Should we see glucose in the urine? No. Proteins? No. What about, can you give me ADH levels? So if we did excess sodium in the bloodstream, what would happen to ADH? More particles, increase in ADH. Good. 500 mils, and we could do this all day, of bicarbonate. So HCO3 water. Whoops. Okay. So we're drinking bicarbonate water. So technically, if we drink bicarbonate water, it's still water and it's not going to dehydrate you. So urine output should go up. Specific gravity, they're inversely related, so it's going to drop. Now, here's a trick. If we have bicarbonate is a pH of about 8, 7 to 8. Okay. So your blood pH is going to be high, right? Your blood pH is going to be 8. And so you're going to hold on to protons instead of secreting them. So you're going to hold on to protons instead of secreting them. And so if you're not peeing out the protons, what's going to happen to pH of urine? It's going to jump up. Good. No glucose, no protein. Am I forgetting something? I don't think so. I think that would be it. What happened if your blood pressure was really low? So let's say, let's do low blood volume. So you have a drop in blood pressure. So what would happen? Okay, so you would release aldosterone. So ALD would be high, right? So would you pee out water or would you hold on to it? Good, you're going to hold on to it so urine output drops. Specific gravity is going to go up. Does this affect pH? Nope. Glucose? No. Protein? No. Okay, good. If urine output is less, what is the color of that urine? Good, it's probably dark yellow. What about if we have diabetes mellitus? So diabetes mellitus, you're gonna pee out glucose in the urine. So, and because you have glucose in the urine, that is gonna pull water towards it. It acts as an osmotic diuretic. If you read your lab, it tells you that. So urine output's gonna go up, specific gravity is gonna drop, but it's actually gonna dehydrate you because you're pulling water from the blood into the urine. It doesn't screw up pH. Depending on how damaged your kidneys are, you may or may not dump out protein. What if you are starving? What we're interested in is, did you dump out proteins? Did you, like under stress, like if I was severely dehydrated, if I was starving, if I was extreme exercising, I'm going to dump out proteins and you may even see albumins. Okay, so that gives you an idea. All right, I want you to spend some time going through the lab. It's really not fun that we didn't get to do this all together as a class and analyze our urine together, but it's really important to be able to look at urine and understand what's going on in the blood of that patient. So if the urine is dark yellow, you know that they're dehydrated, okay? And they are releasing antidiuretic hormone. If they're severely dehydrated, they're releasing aldosterone after that. And if they're releasing aldosterone, what is blood pressure like, right? Blood pressure is really low. And so when you took a patient's blood pressure, it'd be really low. And because it's really low, your heart rate and stroke volume or your cardiac output's trying to compensate for that. So heart rate will be really high. Okay. So if somebody's severely dehydrated, blood pressure drops, heart rate's high, you know that they're trying to pull in water. And so you would give them IV fluids, right? Okay, guys. So go do the lab now and write it out, analyze it, look up normal values, understand how the hormones affect urine output, and regulate homeostasis. Great job, you guys. Great semester. And email me if you have any questions. I'm so happy for you. Yay! Give yourself a pat on the back. Last lecture of the semester. Woohoo! Let's see.